Hello. In this video, I'm going to take you through the viral replication stages of HIV virus. So HIV replication involves several stages. So the first stage is known as binding and entry, or attachment and entry. So in this stage, the HIV virus is going to target the CD4 T cells, which are part of the immune system. The virus attaches to the CD4 receptor and a co-receptor, usually the CCR5 or the CXCR4, which are on the surface of the CD4 cell. It then fuses with the cell membrane. And releases its genetic material into the cell. Once inside the cell, once these genetic materials are inside the cell, the HIV RNA is reverse transcribed into DNA by the enzyme known as the reverse transcriptase. So this HIV RNA is going to be transcribed into DNA, okay, by the reverse transcriptase enzyme. This newly formed viral DNA is then transported into the nuclear cell. So it is going to be transcribed into DNA, and then it is going to be imported into the nuclear cell. What is going to happen next now is that the viral DNA, which has been transported into the nucleus of the cell, is going to be integrated into the host cell's DNA with the help of the enzyme integrase. Okay? Once that happens, once integrated, this viral DNA is going to be called the provirus. It is going to be called the provirus once it is integrated. So the host cell's machinery transcribes the proviral DNA into the messenger RNA. Okay, so there's going to be transcription of this provirus into the messenger RNA, which is then translated into the viral proteins by the host cell's ribosomes. Okay, so there's going to be transcription and translation. So this viral DNA is going to be transcribed into the messenger RNA and then exported out of the nucleus. Then once it is exported, it is going to be translated to the viral proteins. Okay. So once these viral proteins have been made, what is going to happen now is that the newly synthesized viral RNA, the enzymes and the structural proteins, they are going to move to the surface and assemble into a new viral particle. So it is going to assemble these RNA genomes and the viral proteins. They are going to assemble moving to the cell surface. Okay, so these particles are going to be assembled and later they are going to move. So now once that happens, the viral particles, they bud off from the host cell, acquiring an envelope. Okay, so it starts to acquire an envelope and then it has that envelope. So once it acquires an envelope which is derived from the host cell membrane, during this process, the viral protease enzyme cleaves the precursor proteins into their functional forms, leading to the maturation of the viral particles. So once it cleaves off the host's cell membrane, what is going to be there now is there's going to be maturation of the viral proteins. So the viral proteins are those which are inside, so they'll start maturing and then they'll become like this cell. Once they mature, what is going to happen is that the mature viral particles can then infect the new CD4 T cells and hence starting the cycle of infection once more. So this replication process progressively weakens the immune system, leading to the immune deficiency and the onset of AIDS if left untreated. So to summarize, when it comes to the stages, there is attachment and entry, there is reverse transcription, there is integration, there is transcription and translation, there is assembly, and then there is budding and dematuration.